Hello, everyone, and thank you for joining in for this talk entitled Advancing Protein Stabilization Therapeutic Development and Deubiquitinase Targeting Chimera Drug Discovery through Gene and Cell Engineering, presented by Dr. Yongli Shan. Dr. Shan is the Associate Director of Discovery Biology at Vicinitas Therapeutics, where he leads a team of scientists in conducting proof-of-concept studies and exploring protein stabilization mechanisms for target selection. With a strong background in cell biology, molecular biology, and chemical biology, Yongli has coordinated multidisciplinary collaborations and developed genetic and cell-based assay platforms for high-throughput screening. For a complete biography of today's speaker, click their name in the presenter window on your screen. I'm Sean Taylor, and I'll be your moderator for today's event. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the field below and click send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. Dr. Shan, you may now begin your presentation.
Hello, everyone. My name is Yongli Shan, currently serving as the Associate Director and Group Leader of Discovery Biology at Vicinitas Therapeutics. Thanks to Genetically for this opportunity to speak at 3rd Annual Gene and Cell Engineering Virtual Summit. It's my great pleasure to introduce the molecular and cellular platforms of Vicinitas Therapeutics to the audience. At Vicinitas, our vision centers around induced proximity-based therapeutics and our proprietary deubiquitinase targeting chimera or DUBTAC platforms. Deubiquitinases reverse the effects of ubiquitination-dependent protein degradation via proteasome and lysosome by cleaving the peptide or isopeptide bond between ubiquitin and its substrate protein. And a heterobifunctional molecule composed of target protein ligand, a linker, and a deubiquitinase recruiter ligand facilitates the formation of an active ternary complex that leads to deubiquitination of target proteins. Our primary objective is to enhance protein target stability by means of directed deubiquitination, thereby reinstating the normal balance and functional state of proteins. This approach holds great potential for addressing loss of function diseases caused by aberrant protein degradation. The founding stands as a proof of concept of DUBTAC mechanism and selective covalent ligand for deubiquitinase recruitment has been published in Nature Chemical Biology by Vicinitas founders at UC Berkeley. At Vicinitas, we've been building unique cell-based platforms. Genetic and chemogenetic enforced proximity-based vicinity matrix supports the exploratory research for identification and validation of protein targets and stabilization machineries. Vicinity cells are the engineered cell lines and assays employed for drug discovery needs, including ligand finding, induced proximity, and functional readout. Let's talk about vicinity matrix first. Shown on the left is the protein-protein interaction network, which constitutes the gene circuitry driving various biological activities. Induced proximity-based drug discovery aims to establish novel connections between nodes in the graph, generating new protein-protein interactions that offer the therapeutic benefits and functional advantages. At Vicinitas, we are interested in both disease-related target proteins and protein stabilization machineries. To investigate the inducible interactions between protein targets and stabilization machineries, we create a matrix by crossing the list of targets and the list of deubiquitinases. This matrix serves as a high-throughput assay system capable of running hundreds of unique pairings concurrently. The resulting target stabilization and protein abundance change acts as a mimetic of induced proximity by future small molecule DUBTACs. These proof of concept studies play pivotal roles in early discoveries, aiding in target selection and validation. To generate the engineered cell lines carrying edited genes for vicinity matrix and vicinity cells, we employed multiple gene and cell engineering and delivery platforms. These include CRISPR for endogenous gene editing, lentiviral vector for high-efficient bulk stable transfection, and transposon for clonal stable transfection. In contrast to conventional plasmid transfection and passive random insertion, the active integration approaches like lentiviral vector and transposon have unique advantages, including high efficient and better defined genome integration. Moreover, these approaches provide guaranteed integrity of fragment insertion, supported by a nascent quality control mechanism. And of course, many of these materials come from GenScript. To facilitate the selection of target proteins and deubiquitinases, we conducted a comprehensive bioinformatic campaign to assess the expression level, ligandability, activity, and subcellular localization of all the proteins under investigation. On the right side, you can see the collective compilation of deubiquitinases sourced from a publication and public datasets, serving as a DUB encyclopedia. The goal of this bioinformatic campaign is to investigate the bioinformatic proximity between target protein and DUBs as an in silico investigation before diving into the real-world induced proximity. Presented here is an example of our bioinformatic endeavors. We examined the distribution of all identified deubiquitinases across various human tissues from a published proteomic dataset. Building upon the results obtained from our bioinformatic campaign, we employed a series of genetic and chemogenetic approaches to enforce the proximity between target proteins and deubiquitinases. The first generation of these approaches involves an inducible dimerization system called idimerize, which relies on the FKBP, FRB, rapamycin ternary complex. Using the idimerized compound, we effectively mimic a molecular glue that enforces proximity of a target protein and a DUB through dimer A and dimer C tagging. In a homogeneous setup, we combine the idimerize and habit assays to create a matrix in 3 to 4 well plates shown on the lower part by crossing the list of targets and list of deubiquitinases. This plate-based matrix enables high throughput workflow to run hundreds of unique target and DUB pairings concurrently. Based on the resulting target stabilization shown here, we can analyze the fingerprint of either target protein down a column in the matrix or the fingerprint of a DUB across a row. Through this effort, we've identified a few notable super DUBs highlighted in the matrix and many promising hot target proteins that can be stabilized. Moving one step forward, we've introduced the second generation of enforced proximity system, which integrates MATCHIME designs and acts as a closer approximation to the final DUBTAC compound. In this approach, we create bifunctional hybrid compounds by linking small molecule ligands to a target protein and recruiter ligands to well-known protein tags, such as halo tag or DTAG. These compounds mimic the behaviors of DUBTACs and direct the proximity between a target protein and a tagged DUB. The halo tag DUBTAC system replicates a covalent DUB recruiter, while the DTAG DUBTAC system mimics a non-covalent DUB recruiter. In addition to the advantages like a smaller tag size and being duplexable with a halo tag ubiquity nanobrand system, DTAG also enables the controllable and tetratable DUB expression using a validated DTAG-PROTAC based on cereblon, shown on the right. By employing these advanced approaches, we aim to further refine and optimize the induction of proximity between target proteins and DUBs, bringing us closer to the development of effective DUBTAC compounds. Again, the DNA constructs that enables the design of these approaches mostly came from GenScript. Shown here is a schematic presentation of the cell engineering workflow to create enforced proximity matrix. First, we introduce stable extraction of target proteins using a transposon vector on the left. Subsequently, we have the option to either conduct an antibiotic selection and single-cell cloning process, or directly induce expression of the DUB using a lentiviral vector. During this stage, it is essential to maintain the cells with a DTAG cereblon PROTAC to suppress the expression of the DUB, preventing synthetic lethality resulting from the overexpression of two genes. In the final step, we replace the DTAG cereblon PROTAC with a DTAG protein of interest DUBTAC, effectively transitioning the DTAG molecule from a degrader to a recruiter. This modification enables us to trigger and monitor the protein stabilization effects resulting from enforced proximity of the target protein and the DUB pairings. In the life cycle of proof concept studies involving enforced or induced proximity, the next step after observing the induced abundance change is the validation of functional restoration. We need to make sure that we are bringing back functional proteins, not just some junk proteins. In addition to utilizing immediate functional markers as readouts, we are committed to conducting comprehensive functional genomic profiling. This includes transcriptomics and proteomics analysis, which serves as the ultimate means of characterizing the functional effects of enforced proximity or DUBTAC effect. Now let's delve into the details of vicinity cells. Through genetic engineering of the disease-related cell models, we have successfully developed tool cell lines and cell-based assays to support DUBTAC drug discovery and related structure-activity relationship studies. These assays utilize reporter cell lines and operate in heterogeneous systems that can be easily adapted into high-throughput workflows. Besides monitoring the protein abundance change, these assays are able to quantify the intracellular target engagement or ligand binding, ubiquitination or deubiquitination, and ternary complex formation. These mechanism of action studies provide comprehensive understanding of DUBTAC function, guiding lead discovery and optimization of drug candidates. Platform number one is a series of cell lines and assays tailored to detect and quantify the processes of ubiquitination and deubiquitination affecting target proteins. In this platform, we employ various techniques such as labeled ubiquitin antibodies, ubiquitin binding motifs. On the right side, the helo tag the ubiquitin as tracers. These tracers, in combination with the nanoglow habit tag fused to the target protein, establish a nanobright system. By leveraging this system, we can accurately measure the bright ratio, which is a biomass resonance energy transfer, which directly correlates to the ubiquitination level of target protein. This enables us to assess ubiquitination specifically and quantitatively. Platform number one plays a critical role in providing valuable insights into the ubiquitination and deubiquitination dynamics of target proteins and contributing to a comprehensive understanding of their regulatory mechanisms. Platform 2 comprises a collection of cell lines and assays designed for intracellular target engagement, facilitating the discovery of ligands for target proteins and DUBs. These include complementary approaches for the targeted or unbiased ligand binding. For targeted binding, for the targets on DUBs with known ligands, nanobright tracers can be developed. A nanobright system is utilized to assess ligand binding. However, in many cases where we are exploring therapeutics for novel targets or engaging new DUBs without available chemical matters, the intracellular thermal shift assay on the right hand serves as a powerful, unbiased assay for the novel ligand discovery. Platform number 2 offers a comprehensive range of methodologies to investigate ligand interactions within the intracellular environment, enabling the identification of potential ligands for both established and emerging target proteins and DUBs. Platform number 3 consists of a series of cells and assays specifically designed for the detection and quantification of ternary complex formation. This platform plays a crucial role in facilitating the analysis of structure-activity relationship and mechanism of action. To investigate untagged ternary complexes within cell assays, we have employed the alpha screen assay. For tagged proteins in live cells, the nanobright assay is utilized to assess the formation of ternary complex in real time. Platform number 3 provides a comprehensive tool set to investigate and quantify the ternary complex formation between proteins and chemical matters. In this slide, I'd like to showcase the successful application of a hybrid nanobright duplex assay, which enables the simultaneous monitoring of protein abundance and ligand binding. Both assays demonstrated excellent performance, effectively capturing the differences among the compounds within this informant site. This includes their respective interactions with the target protein on the bottom, on the lower part, the binding, and their impact on protein abundance on the higher part, the abundance. For the purpose of de novo ligand discovery, the utilization of a hybrid tagging-based cellular thermal shift assay has proven to be a robust and unbiased intracellular target engagement test. This assay enables the direct measurement of ligand binding and target engagement within the cellular context and has significantly contributed to the characterization of ligands, even in the absence of established SAR knowledge. As a final overview, the last slide and accompanying table provide a summary of the platforms and approaches discussed earlier. These platforms and approaches collectively form an integrated cell-based research and early discovery ecosystem. By leveraging genetically engineered cell lines and utilizing the various methodologies described, these approaches hold significant potential for drug discoveries in diseases such as cancer and monogenic disorders. These conditions often require the restoration of protein stability and function for the therapeutic benefits. Through our innovative research and technological advancement, we aim to make significant contributions to the field of drug discovery and bring about transformative therapeutics. Thank you.
Thank you, Dr. Shan, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. We'll answer as many of your questions in the remaining time. Let's get started. Our first question is, what is the advantage of using genetic and chemogenetic approaches as a proof of concept study for induced proximity-based drug discovery?
Thank you, Sean. That's a great question. So our ultimate goal at Vicinitas is to develop small molecule compound that can enhance protein stability and restore the function. But during the early stage of discovery, when specific chemical matters for target proteins or for stabilization machinery are not readily available, genetic and chemogenetic approaches offer valuable alternatives. So these approaches leverage protein tagging systems that enable us to enforce the proximity of a pairing between a protein target and the ubiquitinase. So several robust systems can serve this purpose, like the constitutive protein complexes system like habit large bait or VHH EGLP antibodies, as well as small molecule inducible systems like the idimerized ternary complex mentioned in the presentation. These protein tagging systems provide fast and straightforward means to attach tags to proteins of interest and create constructs for expressing these tagged proteins in human cells. And through careful design and well-controlled experiment, these approaches effectively mimic a bifunctional DUBTAC, which is the ubiquitinase targeting chimera molecule or all molecule glue stabilizer. So as a result, they can rapidly provide some predictions regarding the feasibility of protein stabilization. Although these genetic or chemogenetic approaches may not fully replicate the complexity of native ligands-based small compound, they can offer a valuable tool site during early stages of research when ligands are not readily accessible.
Interesting. Question 2. Can you please list some of your considerations or criteria for selection of cell lines for POC studies and drug screenings?
Yeah, that's a good question for selecting working cell lines. In our research, we have several criteria for consideration, and I would say target selection being the most crucial one. To aid in the target selection process, we rely on bioinformatic databases like CCLE, TCGA, and clinical variants for oncological targets. And our primary focus centers on monogenic diseases and associated target genes. So that will allow us to delve into specific MOA of these conditions to focus on one gene. To study the protein functions mechanism of action, we leverage cell lines carrying loss of function mutations. And this approach provides insights into the consequence of reduced or absent or aberrantly degraded protein. That will enhance our understanding of disease pathways. For investigating protein stabilization, haploinsufficiency offers a straightforward strategy. So we will examine the cell lines with insufficient functional gene copies, and we can gain critical information about protein regulation and through stabilization, like if we can increase the abundance of these haploinsufficient genes, what function output we can get, and precisely monitor endogenous protein abundance, we employ immunoassays for endogenous detection or habit knocking techniques for tagged detection. And these methods enable accurate tracking of protein levels within these cell lines. So for cell lines with a loss of heterozygosity that will have non-functional copy of the target genes, we also develop an approach to reintroduce open reading frame cassette and to restore protein expression and protein function. This will give us a unique opportunity to explore the consequence of protein recovery in disease context and in the context of enforced proximity matrix experiments involving overexpression of a list of targets and the combination of a list of ubiquitinases, we opt for a universal cell line like HEK cells. These cells are easy to transfect and exhibit decent protein expression. That will facilitate the experiment precise to create the proximity matrix. And additionally, to streamline our workflow and enhance efficiency in high throughput experiments using 3 to 4 well plate formats, we utilize suspension cell lines. So these cell lines behave like liquid reagents. You just need to do the pipetting. That will make the pipetting more efficient and further improving the experiment throughput. Although it's kind of a small improvement in the concept, but it's a very crucial improvement in the experiment workflow.
And just on that thought, because you mentioned you're working in 3D 4 well plates.
Yes.
There's actually enough cells in those plates to get a good readout from the nanoparticles in there?
Yeah, that's a good question. So for 3 to 4 well plates, we have sensitive plate readers, like a few models like from TCAN or from Invasion that have ultra-sensitive bioluminescence readout. And also the bioluminescence readout based on nanolog or luciferase has a very good performance in signal background ratio. So the signal is very specific, and the background is low. Yeah, and we have a suspension cell line that has very decent expression after transfection, and with the ultra-sensitive habit detection, the signal background ratio is very great.
Very good, very good. It's great that you're able to have that to be able to do things in such high throughput. That's awesome.
Exactly. Yeah.
The next question is, why do you choose lentiviral vector and transposon systems as the two primary gene delivery methods?
Lentivirus and transposon. Yeah, that's a good question. So traditional people are using DNA plasmid and random insertion to achieve stable expression. So in contrast to the random insertion and low efficiency, often associated with the conventional stable transfection using DNA plasmid, lentiviral vectors and transposon offer highly efficient gene delivery to introduce exogenous fragments into the genomic DNA. So lentivirus is known for its unparalleled transfection efficiency, serves as a robust block transfection method in our platforms. That leads to stable expression of target genes upon completion of the reverse transcription and the genomic integration. However, the packaging capacity of lentiviral vectors are restricted. That restrict the insertion size to only a couple of kilobases, and restricted to only a subset of small and medium-sized open reading frames. On the other hand, transposons has a significantly higher carrying capacity, or even like unlimited capacity in most cases. It utilizes an active cut and paste mechanism to integrate DNA fragments into genomic DNA. Nonetheless, the initial step for transposon still relies on conventional transfection methods like liposomal electroporation. Both lentiviral vectors and transposons exhibit some degree of precision in the genomic integration site preference. They still fall short of the precision achieved by CRISPR-based approaches. A notable advantage shared by both lentiviral vectors and transposons is their role as a nascent quality control mechanism, because their life cycle of transfection guarantees the integrity of the fragment. Plasmid transfection is lack of this guarantee, and sometimes we may experience loss of random regions from the fragment. That will potentially impact the reliability of experiment results.
Sounds like we need a lentiviral vector that can insert much larger fragments. Maybe that's coming in the pipeline at some stage.
That will be an ideal solution to all these problems.
Absolutely. The next question, and I think we basically have time for one more question here, and I'm just trying to, is what is the rationale of protein stabilization-based therapeutics, and how is it compared to protein degradation modalities?
Yeah, so basically that's a question about DUBTAC versus PROTAC. They are both based on ubiquitin system that act as a protein quality control and degradation system. So it's based on ubiquitination. So ubiquitin is a small regulatory protein, and through the process known as ubiquitination, ubiquitin molecules are covalently attached to target proteins, marking them for degradation by the proteasome system. Deubiquitinases. So the ubiquitination is mediated by E1, E2, E3 ubiquitin activation, conjugation, and ligation pathway. Deubiquitinases reverse the effects of ubiquitination, so it will reverse the function of protein degradation through proteasome and lysosome by cleaving off the ubiquitin peptide or peptide bond between ubiquitin and its substrate protein. Hyperbifunctional molecule composed of target protein ligand, a linker, and deubiquitinase recruiter ligand will make a DUBTAC that can facilitate the formation of a active ternary complex that leads to deubiquitination, and then restore the abundance of target protein through increasing the protein stability. So in comparison to protein degradation-based PROTAC approach, PROTAC is to remove unwanted, sometimes harmful or toxic proteins through ubiquitination and proteasome. Vicinitas properly, the DUBTAC approach aims to restore the levels of aberrantly degraded proteins. The aberrant degradation will cause diseases, and by reverse that negative effect, we can confer therapeutic benefits through protein stabilization and reverse the aberrant degradation.
That's fascinating. Really interesting work. Just before we close, Dr. Shan, do you have any final comments for the audience? Just a wrap-up comment, a few words.
Yes, sure. So protein stabilization is still at the frontier of protein homeostasis-based therapeutics. We don't have tool compounds like protect person that can enjoy. There are still a lot of unanswered questions. So early discovery plays a pivotal role in development of this area. It's an exciting area to work on, and there are a lot of biology, also some biochemistry campaigns to support and to collaborate to make this field moving forward.
Fascinating discussion. Really interesting work, Dr. Shan. So before we go, I'd like to thank the audience for joining us today and for their interesting questions. And once again, a big thank you to Dr. Shan for joining us today. Until next time, have a wonderful day, everybody.
Thank you so much.